We are now going to begin the section of notes discussing cell basics. This should be on page 4 of your notes. At the top it says cell theory, so please find that section and be ready to go. So the cell theory has 3 main ideas. First, all organisms are composed of one or more cells. Second, the cell is the basic unit of organization of all living things. What this means is that cells come together to make tissues, tissues come together to make organs, organs come together to make organ systems come together to make all living things. So that's why it says it's the basic unit of organization. And last, all cells come from existing living cells. You could have also said other living cells. Okay, this is just the idea that cells just don't poof come out of nowhere. They have to be dividing from another cell. Now, the cell theory is important because it describes the basic similarities among all living things, whether it is a single-celled protist or a multicellular organism like a human. We all have some similarities because of the cell theory. The other important thing is that it helps us to compare and make predictions about organisms, because we can look at what things have done in the past and what things are doing now. Then we can talk about what those organisms may do in the future. Now, just some cell basics. The size, the shape, and the organization about the cells. First off, the size. The cells are usually measured in what's called micrometers or micrometers, whichever way you want to say it. And it's got that funny little symbol there that kind of looks like a messed up P/U and an M. Okay, 1 micrometer is 1/1,000,000th of a meter. Okay, so cells range from anywhere from 1,000 micrometers to less than 1. And this image here just kind of gives you an idea of how small these micrometers are. And yes, that is correct. A micron is also another word for a micrometer. So let's look at the shape now. Cells can be round, square, rods, spirals, etc. They can be all kinds of shapes. The organization of those can be either unicellular or multicellular. So if you look over there on the right hand side, we've got some different shapes, and then you can also see how those shapes can combine together to be like grouped. But we have bacillus, which is kind of a rod, coccus, which means that it's more of a sphere shape. Diplococcus means 2 of them. Streptococcus means that they're all kind of in a line and almost like a strip. Staphylococcus is bundled, okay? Vibrio is kind of a whip-like, and Spirillum is more of a corkscrew. That picture for Spirillum doesn't really do it justice, but imagine like a telephone cord, something kind of like that. Okay, and then down there at the bottom it shows an example of E. coli, which is a single-celled organism, and a dog, which is multicellular. Okay, for all cells, you need to know that they are surrounded by a cell membrane. Yes, all cells, including plant cells. The cell membrane helps maintain the cell shape, and it controls the transport of molecules into and out of the cell. Now we're going to spend a lot more time in the next unit talking about how exactly this happens. Right now you just need to know that all cells have this cell membrane, and their job is to control what goes in and what comes out of the cell. All cells also have cytoplasm, which is a jelly-like fluid that makes up most of the cell, and it kind of allows all the other cell parts to kind of float around and just be suspended. All cells also have ribosomes, which are structures that synthesize proteins. Now I want you to make a little side note for synthesize. Synthesize means to make. Okay, so the job of the ribosomes is to make proteins, and all cells, plant cells, animal cells, all kinds, have ribosomes. Cells are divided into 2 groups based on the presence of a nucleus. A prokaryotic cell means that it has no nucleus. Remember, no. We talked about this in the first unit. The DNA is just in a circle, and it is a simple structure. So here's an example here of a bacteria. The bacteria, as you can see, has DNA in the center, but there's no circle around it showing that there's a nucleus bounding that DNA to one spot. It's just kind of floating there in the middle. The other kind of cells that we have are eukaryotic cells. Eukaryotic cells have a nucleus, and that nucleus contains the DNA and acts as the cell's control center. It's kind of like the main computer system. These eukaryotic cells, in addition to having a nucleus, will have membrane-bound organelles, which are specialized compartments that do specific jobs. This is what we've been talking about all week. These are the things like the nucleus, the chloroplast, etc. These are a very complex structure, these eukaryotic cells. So the prokaryotic ones were very simple. They don't have a lot about them. They're just pretty much just holding the DNA and some ribosomes. But the eukaryotic cells, on the other hand, are almost like a city. They have all these things going on inside of them. They're very complex. So some examples of eukaryotic cells include anything from Protista, fungi, kingdom Plantae, and kingdom Animalia. So let's kind of talk about comparing prokaryotic and eukaryotic cells. You've got a Venn diagram there, and I want you to place each of these words in the appropriate spot. Okay, so put prokaryotic on the left-hand side, eukaryotic on the right-hand side, and what's included in both. So, no nucleus, where should that go? No on the prokaryotic side. Nucleus, obviously, goes on the eukaryotic side. Bacteria are prokaryotic. Cells, think about this one. Where would cells fit? In both. Very good. No membrane-bound organelles. That's going to go on the prokaryotic side. Remember, they're very simple. Cell membrane, think hard. Yes, that is found in both. Protista, fungi, Plantae, and Animalia would be the eukaryotic. Membrane-bound organelles are also found in the eukaryotic cells. Ribosomes, found in both. Cytoplasm, also found in both. Simple structure, prokaryotic. Complex structure, eukaryotic. DNA, both. The next section of notes is kind of odd. I'll admit it's a little bit confusing only because we try to make it harder than it really is. This is just saying that you need to be able to recognize that a model is different than the real thing, and that there are some models that are better than others, whether that model be a 3D model or just a diagram on your paper. You just need to be able to assess that to be able to say this is good for this reason, this is bad for this reason. So I want you to just look over these criteria here and kind of study them, seeing what you're supposed to look for. And then I want you to look at the two cells on the next page that I show you, and I want you to fill in your notes just some brief comments about each of those criteria. Okay, so you should pause the video at this time, take a look at these, fill in your notes. We'll be going over them briefly at the beginning of class.